Unit 1 Foundations Biological Hazards. Every week or so, it seems, we hear about another outbreak of a foodborne illness caused by tiny organisms that cause disease called pathogens. Sometimes the outbreak involves a dozen people, and sometimes hundreds. Sometimes people die. Food producers, processors, and manufacturers play a key role in keeping the U.S. food supply, and so do those who work with them. In order to understand how and why pathogens threaten our food system and how our food can be protected, it is necessary to have a working knowledge of biological hazards. We'll start with some relevant terms that will be repeated over and over again during this course. The first term is biological hazard or biohazard, which means a biological substance that poses a threat to the health of animals or humans. These can include bacteria, viruses, and toxins from living organisms. The next term is adulteration. The Federal Food, Drug, and Cosmetic (FD&C) Act of 1938 defines adulteration very specifically. Food is adulterated if it bears or contains poisonous or deleterious substance which may render it injurious to health. This includes, but is not limited to, unsafe pesticides, unsafe food additives, diseased animals, and filthy, putrid, or decomposed substances. A food is also considered adulterated under the law if it has been prepared, packed, or held under unsanitary conditions, or subjected to unregulated radiation. 21 U.S. Code Section 342. The next term is foodborne illness. A foodborne illness results from the ingestion of a pathogen or chemical or biological toxin. 3 distinct pathogenic models classify foodborne illness. These are the infection model, the toxico-infection model, and the intoxication model. Infection is the result of pathogens entering the body and multiplying in the intestines. In the toxico-infection model, the body is also invaded by a pathogen, and the toxin produced by the bacteria causes the illness. In the intoxication model, a person eats a food that is contaminated by the preformed toxin, which was not produced within the body. Each pathogen causes a unique set of diseases, but the common initial symptoms for all 3 models may include fever, nausea, vomiting, abdominal cramps, and diarrhea. A susceptible population includes the YOPI populations, otherwise known as the young, under 5, old, over 60, pregnant, and immunocompromised populations. Young children are more susceptible to some biological hazards because their immune systems are not fully developed. Examples of immunocompromised populations include patients who have undergone organ transplants or chemotherapy, have AIDS or another illness that compromises the immune system, or have inherited a disease that damaged their immune system. Because their immune systems are damaged or weakened, their ability to fight infection is decreased. Pathogens are all around us, and even those with compromised immune systems don't always get sick. The threshold inoculum, or infectious dose, is considered to be the minimum quantity of a pathogen that is needed to cause infection. This estimate can differ drastically between both organisms and individual patients. For example, the bacteria Escherichia coli 0157:H7 has a minuscule infective dose of just 10 to 100 cells, while Bacillus cereus requires an infective dose of 100,000 to 100 million cells, 10 to the 5th power to 10 to the 8th. A reportable diagnosis or disease is a diagnosis that is considered to have public health significance. Doctors, clinics, and laboratories must report these diseases when they are diagnosed to either state and local health departments or the Center for Disease Control, CDC. They are also referred to as notifiable diseases. The National Notifiable Diseases Surveillance System, NNDSS, allows all level of public health, local, state, territorial, federal, and international, to share information about foodborne diseases, which helps officials to identify outbreaks of disease. All foodborne illnesses are designated as reportable. 5 cases of food poisoning in Tennessee might go unnoticed, but when there are 10 or 15 more cases scattered around in surrounding states, inspectors and investigators start looking for a common source. All foodborne diseases are reportable because they may be communicable or may indicate a problem in the food or water supply. Initial symptoms of a foodborne illness usually last from 24 hours to less than a week, but continued infection often leads to complications and serious illness. Examples include the development of Guillain-Barré syndrome after a Campylobacter infection, the hemolytic uremic syndrome (HUS) after infection by Escherichia coli 0157, or severe liver damage after chronic exposure to aflatoxin. Biological hazards have an enormous effect on the U.S. food system. Some of the effects are socioeconomic. Foodborne illness and disease have been considered a public health concern since the early 20th century, and recent studies attribute some 6 million cases, 350,000 hospitalizations, and 9,000 deaths to foodborne infections each year within the United States. Biological hazards also have an enormous financial impact on the food industry. The financial burden of biological hazards can be exemplified by a 2013 research study on Shiga toxin producing Escherichia coli, STEC. This group of pathogens costs the food industry $456 million annually, with $405 million attributed to E. coli 0157 alone. Marks, Tohemi, and Sui, 2013, Scallon, et al., 2011. Because of the risk that biological hazards pose to the food system, the notoriety of various cases and concern about food poisoning has led in the past to the creation of regulatory bodies including the Food and Drug Administration, FDA, and the United States Department of Agriculture, USDA. These agencies investigate and fund research on current and emerging pathogens as well as conducting screening and detection for pathogens in food products. Federal regulations dictate management and enforce processing requirements used to reduce pathogens within the food industry. In food production, however, it has been and will continue to be the food industry's responsibility and burden to reduce the prevalence of foodborne disease while creating quality, wholesome products. It is the industry's responsibility to ensure that processing parameters and product formulation meet federal standards and that the product emphasis is on food safety during production, storage, and distribution. While only pathogens are actually harmful, food processors and manufacturers also need to be aware of spoilage organisms. Pathogens will cause illness or disease via infection, toxicoinfection, or intoxication, and the illness may result in hospitalization or death. Despite their serious effects, pathogens might not affect the sensory or aesthetic qualities of a product. Spoilage organisms, by contrast, produce off flavors, odors, textures, and appearance. They change the sensory or aesthetic qualities of a food, rendering it undesirable. Spoilage organisms may also indicate the pathogens in food or feed or may change product conditions, allowing pathogens to grow. There are many sites online to learn more about biological hazards and their impacts. The Food and Drug Administration, the U.S. Department of Agriculture, and its Food Safety and Inspection Service are both good sources, as are the Centers for Disease Control and public health agencies. Here are their website URLs. Finally, your local university extension personnel are an excellent resource. In 1993, a widespread outbreak of foodborne illness caused by undercooked hamburgers served at the Jack in the Box fast food chain on the West Coast led to the deaths of several children and dramatic changes to public policy. The industry changed in significant ways, including E. coli 0157:H7 was upgraded to become a reportable disease by all state health departments. FDA increased the recommended internal temperature for cooked hamburgers from 140 degrees Fahrenheit to 155 degrees Fahrenheit. The USDA's Food and Safety Inspection Service, FSIS, introduced safe food handling labels for packaged raw meat and poultry sold in retail markets. FSIS introduced testing for E. coli 0157:H7 in ground meat. USDA reclassified E. coli 0157:H7 as an adulterant in ground beef. USDA introduced the Pathogen Reduction and Hazard Analysis and Critical Control Point PRHACCP program. The National Cattlemen's Beef Association created a task force to fund research into the reduction of E. coli 0157:H7 in cattle and slaughterhouses. Jack in the Box completely overhauled and restructured its corporate operations around food safety priorities, setting new standards across the entire fast food industry. The food industry utilizes sampling in several ways to detect harmful microorganisms. What we call surveillance sampling can be either routine for regulatory reasons or in response to an epidemic. Routine sampling can be used for quality control and evaluation of processing procedures to reduce pathogens or spoilage organisms. Environmental sampling, for example, is carried out to determine the presence of pathogens on food contact surfaces of equipment and on non-food contact areas in the facility. Microbial sampling of feed and feed ingredients is also useful in detecting Salmonella and E. coli 0157:H7. Product sampling provides a snapshot or one-time reading of the condition of a product at a given point in time, so the more frequent the sampling, the better the monitoring of pathogens. Since continuous laboratory monitoring of all product is not possible, continual process monitoring of the temperature, pH, or pressure, for example, is a much more effective control than monitoring product for biological contamination. Sampling to validate process technology involves measuring the level of the biological hazard before and after the treatment being tested to see if the hazard is reduced to the expected and acceptable level. Regulatory sampling is sampling that is required by federal agencies as a way to detect key pathogens during production. For example, raw ground beef is always tested for E. coli STEC, and poultry is tested for Salmonella. Epidemiological sampling leverages public health agencies and hospitals to report the prevalence of foodborne illness and disease within a population. For cause sampling is sampling that takes place for a specific reason instead of being routine. In response to an outbreak of foodborne illness, for example, investigating bodies will do outbreak sampling. They will collect samples from outbreak victims and potential sources in an effort to identify the causative agent. Research sampling or validation studies may use what we call event-based sampling to answer research objective or validate processing technologies. There are many human illnesses caused by biological hazards. Zoonotic diseases are diseases that can actually be transmitted between animals and humans in various ways. Examples of pathogens that can be passed from animals to humans include Escherichia coli 0157:H7, Staphylococcus aureus, Salmonella enteriditis, Campylobacter jejuni, and Yersinia enterocolitica. As described when we talked about the Jack in the Box foodborne illness outbreak, E. coli in hamburger can cause serious illness if the hamburger is undercooked. Salmonella in raw or undercooked poultry can cause an illness called salmonellosis. Campylobacteriosis, caused by Campylobacter jejuni, is among the most common of the foodborne illnesses worldwide. Cattle, swine, and poultry all may carry C. jejuni, with the most common routes of infection being unpasteurized milk or undercooked poultry. Yersiniosis, caused by Yersinia enterocolitica, is most likely to occur in young children and can also be transmitted by drinking unpasteurized milk or by eating undercooked meat products, particularly pork. Pathogens can also cause illness in food animals. A very serious example of an illness caused by a biological hazard is bovine spongiform encephalopathy, BSE, or mad cow disease, in cattle. There is a strong link between BSE and a devastating disease known as Creutzfeldt-Jakob disease, VCJD, which occurs in humans. Both are caused by prions, which you will learn about in Unit 2. Another example is foot and mouth disease, FMD, a viral disease that causes blisters in the mouth and on the feet of ruminant animals such as cattle. FMD can cause a permanent decrease in milk yield and lameness. Some food animal diseases have the potential to seriously impact our food supply. One example is avian flu, the result of an infection with the type A strain of the influenza virus. This strain is highly pathogenic with mortality in poultry reaching nearly 100%.